I guess let me take a step back. You dissect the umbilical cord and placenta, you start centrifuging, you're separating the tissue from the cells with collagenase, and then you're taking that and spinning it further, what's called the pellet. This little white pellet you put in there from centrifugation, you put that in your flask, you add the culturing medium, it starts replicating, the MSCs start growing, and they adhere, like let's pretend this is the surface of the flask, they adhere to it, they stick to it. It's like an ivy growing up a house. They hold on, where the other cells are free bound in the fluid. So after passage one, and all that means is that they've eaten through the culturing medium. So now you're left with MSCs and whatever residual cells are left. We do what's called an enzyme flush. Put in this specialized enzyme solution, it flushes that out. And now you've got MSCs, but one passage doesn't clear it completely. It's just like washing a dish in your sink. You're going to have some left over. So now you go to passage two, do the same thing. Passage three, by then you've got pure isolated mesenchymal stem cells. And then we go two more past that. So the whole passage thing and all this and people using that as an argument's ridiculous. What matters, and I should have got to this quicker, is not just the quality control, but how it's processed and the sterility. I've seen labs, or people call labs, where it's just a hood. In a hood, look, a hood is, it looks like this, it comes up over and it filters air and you work within it. That's where the biologists work, that's where they process. So we're dissecting the umbilical cord in that, they're inside the hood because it's called rated ISO 5. ISO stands for International Standards of Operation. So for the whole world, it's the same or not regulations, but standards. Right? And biology, these sort of labs have very high standards. The highest standards are actually for aerospace. My dad had a big aerospace company, and geez, you should have seen what the ISO ratings inspections took. It's nuts. And what the way they measure it, the main thing is the particles in the air. So you got to make sure no particles, because that's what can lead to infection or other problems. This is medical. We need to make sure it's as sterile as possible. And there's clinics and labs out there that have no sterility. They just have a hood. It'd be like in a room like this with a hood. They're like, oh, it's ISO 5, which ISO 5 is awesome. But if the room you're in is not ISO rated and it's just an open room with the, fan in it, well, that's going to be filled with particles. That's just nature. There's stuff floating all around you can't see. So with our lab, you enter ISO 9. Then the changing room, you're looking at ISO 8. Then you go through an air shower, blows all this air on you and sucks it through a HEPA filter, what's blown, so any particles get sucked off. You're all in this big spacesuit looking thing going in, and now you enter ISO 7. So our lab, the whole lab is ISO 7, and then our biologists work in an ISO 5